My passion remains tuberculosis, and it's amazing that now herpes has entered into it. I want to tell you about pursuing Edward Jenner's revenge. Now this is a real old paper, 1798. And in fact, you know, Edward Jenner was doing this process of variolation that had been started in China in the 10th century. And you know, people in China had found that, you know, if you went and you took a lesion from somebody who had died of smallpox, or who was dying of smallpox, I mean, they had no treatment for it. They found if they took the lesion, they scraped it on the arm of the person, they'd get sick, but they wouldn't die of smallpox. And that was the initiation of variolation. And Edward had made the observation that the milkmaids that often got cowpox would never ever get smallpox. So in fact, he took, Sarah used to do the milking of Blossom here, and she got cowpox, and he took a lesion, stuck it in the gardener's son. He had flu-like symptoms for a week. Then 3 weeks later, he challenged him with variolation, and the kid had no lesions whatsoever. And that was our first vaccine. And the word vaccination comes from the Latin word vaci for cow, vacca for cow. You know, it was just sequenced a couple of months ago, the virus, and it actually turns out to be a horse virus. So it should have been called equination. If you don't, I mean, most of the students in the room aren't even vaccinated for smallpox. It was eradicated from the planet in 1980. What I find amazing was in the century that smallpox was eradicated, 300 million people died of smallpox. Just showing you the power of vaccines. The reason why I call this Edward Jenner's revenge was that his wife and son did not die of smallpox, but they died of tuberculosis. So this is for Eddie. You know, in 1815, when they died, you know, we did not know really what the cause of tuberculosis was. Many German scientists were convinced that it was a cancer. And it was really the pioneering work of Hansen that first stained Mycobacterium leprae, combined with Jean-Antoine Villemin, that actually distinguished. He took tumors from dead soldiers and transferred them to rabbits and showed that they never ever got any disease. But when he took tubercles from soldiers that died of consumption, tuberculosis back then, all the rabbits got tuberculosis. And Robert Koch knew these papers, and in 1882 he wrote the following postulate. He said, to prove that tuberculosis was caused by invasion and multiplication of bacilli, it was necessary to isolate the bacilli, grow it in pure culture, then transfer the infection. And for the first time, when we knew the cause of tuberculosis, we could go back and think about making a diagnostic test. You know, Zeal and Nielsen, they never knew each other, improved the Gram stain, where they showed if they added acid alcohol, you know, to the fusion, alcohol would take away the dye from a Gram negative, whereas Gram positives would retain it. But if you added acid alcohol, it took away the dye from the Gram positives, but Mycobacteria retained it. When we knew the cause, that we knew if it was being spread, we could implement different public health measures. Edward Livingston Trudeau started a sanatorium up at Saranac Lake. People used to have spittoons to kind of control the disease. You think about it, even though it was discovered in 1882, we didn't get a vaccine, BCG, till 1921. And we didn't get a drug till 1943. And I point this out that, you know, there's got to be a commitment to basic research, knowing that eventually things are going to pay off. Now, despite having, I mean, look at TB today. Over 1.6 million people died of this disease last year. Over 10 million new cases. And now we've seen the emergence of strains that are resistant to all 10 drugs that we use to treat TB. And this is happening despite the availability of a vaccine called BCG and a sterilizing chemotherapy. So my history here at Einstein started with Barry Bloom. Barry realized that BCG, something he had worked with for his doctoral work, was the only live vaccine that's given to children at birth. And he was very involved with the World Health Organization and the various diseases that children face in the developing world. And he said, Bill, you know, when I first met him, he said, if you could come to my lab and figure out how to put DNA into BCG, now we'd be able to go ahead and maybe make a vaccine against TB and malaria, TB and HIV, TB and herpes. And so that was the reason I came here. You know, for my PhD, that's me as a graduate student. And you can see I'm holding my growth media. I had the privilege in Roy Curtis's and Josie Clark Curtis's lab to grow the leprosy bacillus in 9-banded armadillos. And it used to take 2 years to grow it up. And I got to make the first genomic libraries of the leprosy bacillus. And Barry knew of me, and he told me, why don't you come and work on developing BCG as a vaccine vector? I knew from being trained by Roy Curtis and Stanley Falco that in fact, not only was genetics going to be important, moving DNA going to be important for making these vaccines, but it was important in gaining basic knowledge. You know, when I started in '85 here at Einstein, we didn't know the target of isoniazid or any TB-specific drug. We didn't know the basis of acid-fast staining. We didn't even know if we could isolate an oxytrophic mutant. We didn't know any of the genes involved in persistence. And we did not know why BCG was attenuated. Now you may find that shocking, but how do we know stuff? The way that we know the cause, you know, of infectious disease is fulfilling Koch's postulate. In an analogous fashion, the way that we know the cause of a genotype is by fulfilling molecular Koch's postulate. To prove that a phenotype is caused by a genotype, you have to have a mutant, you have to clone the gene, and you have to transfer it. That is the essence of genetics. And the problem with TB was nobody had ever figured out how to move the DNA into TB. And when I came here, I decided I was going to go ahead and work on viruses and infect mycobacteria. But I didn't have any. So I used to live up here, right up the street on 2031 Paulding. And I just took dirt from my backyard back in July of 1985, and I played it out on M. smegmatis, and I got these plaques. I named it affectionately BXB1, the Bronx Bomber. You know what? There's 53 papers on Medline this morning where the Bronx Bomber integrase is the most efficient integrase that has been found in nature. And people use it to make transgenic plasmodium. I used it to make transgenic plasmodium with David Fidduck. Other people, Arabidopsis, Drosophila, mice, human stem cells. And in fact, that phage hunting, which I won't talk about today, has led to an international program where my twin sister and Graham Hatfull, who came to my lab to sequence phages, now have a program taught in 142 institutions around the world. And over 10,000 students have taken this course and isolated phages. And we have 2,500 sequenced genomes of mycobacteriophages today. Using phages, we systematically developed all the tools so we could genetically manipulate TB. Now, I want to point out that using a technique called specialized transduction, we're about to knock out every gene in TB efficiently using this phage. And we're going to be doing that, and that's an ongoing project in the Jacobs lab. Michelle Larson is helping on that. The point about this is having the ability to transfer genes allowed us to figure out why mycobacteria stain acid fast. Even Robert Koch himself said, you know what, I see a lot of cells that aren't always acid fast. But it became the test. And you know what, we finally figured out, Robert Koch would love this, we've now figured out that in fact acid fastness is regulated by a signal transduction pathway. And in many TB infections, there are acid fast negative TB that we can't even see. We identified, the target of isoniazid with John Blanchard and Jim Saccatini. We figured out the molecular mechanism of how it kills the cells. We went on and isolated oxytrophic mutants, which are powerful tools. And Michael Bernet and Sangeeta Tiwari have recently gone back and shown that, in fact, when you starve bugs for methionine or arginine, it's actually sterilizing. We've gone on and identified new genes that are involved in persistence. Let me explain that a little bit further. Because in a lot of ways, what relates herpes and TB are these latent infections. So the point is that the knowledge has allowed us to make new diagnostic tests. It has allowed us to approach rational drug design, but then even study the process of death of a TB cell more carefully. So with John Blanchard and Jim Saccatini, we know now, down to the hydrogen bond, how isoniazid inhibits an enol reductase that leads to the lysis of the TB cell. But what's amazing that we study now in the lab all the time, and Catherine Vilches set this up, nobody had done this before. Catherine just started to take cells after she had isoniazid daily and counted the number of bacteria on the plates. And what she discovered was amazing, that at day 3, she killed 99.9% of the bugs, but she couldn't sterilize them. Short course chemotherapy for TB is 6 months long. Why? Because of these persisters. They are not genetically resistant. They are phenotypically resistant. And we know this because Brian Weinrich went on and isolated the cells and did microarrays, and we find a unique stress regulon that is turned on in these cells. So what this tells me is that a population of TB is a heterogeneous population. And we know that they turn on these stress responses. They don't have mutations at this point. And TB, Lena Kaminsky in our group has gone on and pursued this further by knowing that to get this unique RNA transcriptional profile, TB has 13 different sigma factors. She knocked out all 12 of the non-essential, and she's found one that sterilizes, Sig-E. What's very cool is for diagnostic purposes, we've made these phages that can make TB light up in hours. Take this phage that has this screaming hot GFP gene and add to it an RFP gene, but that's driven by a persister promoter. This is a 3-day movie of TB where we put it down on a microfluidic chip. We add the phage and isonize it, and you can see initially all the cells are green, yellow, and then the yellow cells die, and at day 3 you're left with red cells. Those are persisters. We can visualize them for the first time. What's so cool is Catherine Vilches, by pursuing an isonized resistance mechanism, found that a reducing agent got rid of persisters. So she went back and she added vitamin C with isoniazid. Here's the regular isoniazid. And she saw when she added vitamin C with isoniazid, it sterilized the culture. In fact, vitamin C by itself, if you add it at high enough concentrations, that's 4 millimolar, will actually sterilize XDR TB. Now 4 millimolar is the only way you can get that in the body is Linus Pauling's IV injections. We can't get it orally. But at lower concentrations, we actually stimulate respiration and we don't form persisters. And Catherine's just recently discovered when she adds vitamin C to the 4 drugs we use to treat TB, she can sterilize instead of 20 days in 6 days. The point about this is basic knowledge is going to get us to the cure for TB. So let's come back to vaccines. Here's Dr. Calmette and Guerin. So the TB vaccine is Bacille, Calmette, and Guerin. You know, we found the primary attenuating mutation, COSU in my lab, went back and deleted the genes, and we could complement it. And the point about that is now we know it's a specific secretion system that is knocked out. And we thought, well, maybe we could protect better if we went back and made the mutants. And I just have to tell you that I have a long ongoing collaboration with Steve Porcelli and John Chan to make better TB vaccines. And every mutant that we've made, even when we went back and remade the BCG mutant from virulent TB strains, I mean, here's uninfected, here's BCG. The best mutants that we've isolated protect a little bit better, but they don't sterilize. They're just, everybody's efforts to this point in time have not given us a better TB vaccine. We really need something different. Roy Curtis, my mentor in the first week in the lab, he asked me, is it better be smart or lucky? And of course, the correct answer is both. And Louis Pasteur basically summarized that. They said chance favors the prepared mind. It was Louis Pasteur who made the first rational vaccine. He was trying to beat Koch to fulfilling this postulate. And he had Vibrio cholera, a Vibrio that killed chickens. And he showed, first time he did the experiment, he could kill all the chickens. He was all super excited, and we repeated the experiment, and it didn't kill the chickens. And what he ended up discovering, serendipitously, was that as he passaged that strain in vitro, it became a mutant. The mutant outgrew the wild type, and so he had an attenuated strain. But he was smart enough to realize, well, they must be protected. And so he went back and he challenged them, and he showed now that they could be protected. So why did I start working on herpes? Part of it is, as Alan says, I go to Durban. And what's common to Durban and the Bronx is we have a lot of STDs, and HSV2 is a big problem in Durban. It's also the epicenter of the TB HIV epidemic, and that's why we went there. But in that process, I had the great fortune when Betsy Harold was being recruited here. She was telling me about herpes. and telling me how we needed a vaccine. And I thought, well, let's try some recombinant mycobacteria. This will be different. And as she mentioned, you know, we tried that. But I was really attracted to it too because, you know, typically when we do a virulence experiment in mice with TB, it takes a year to kill mice. And here with herpes, we're able to kill them in 9 days. I thought I died and went to heaven. But here's Pablo, you know, and Pablo and Chris had gone ahead and made this mutant. Pablo made the mutant. But, you know, for 3.5 years, they had cloned these antigens in BCG. And we would mix and match, and every 3 months they'd challenge. They killed every mouse. It was only because Betsy had, you know, Betsy had decided to make this mutant. As Betsy pointed out, now we have a vaccine that I am convinced elicits sterilizing immunity. You know, at 6 or 7 days post-challenge, in any mouse or any guinea pig we've challenged, we cannot find a challenge virus. So now, let's change our vision. If you ever go to a safari in Africa, you know, they tell you to look for these 5 animals. I haven't seen them all, but I know they're out there. But my vision now for Africa is different. My big 5 are these. That we want to make vaccines that are going to prevent, like Barry Bloom had hoped, these diseases that we see in the developing world. And now we have a very different vaccine vector. So our hypothesis is that we have a vaccine vector that selectively induces ADCC antibodies. I know of no other vaccine platform that does this. So now the idea would be that we could go ahead and make vaccines that elicit ADCC antibodies. And you know, since in the last few years, there are lots of people thinking about ADCC antibodies and finding that this is the primary correlate of protection in vaccines, not neutralizing antibodies. So our idea was that we could go ahead, and a very talented student, Joe Dardick, with Kayla Weiss, said, why don't we go back and we want to see if we can get protection in the lung for the TB vaccine. Let's go see if we can make a flu vaccine. And so the idea would be to take the gene, the hemagglutinin, and clone it into the herpes. And so we've made this wonderful red virus that we can replace, and it becomes clear now. And Joe's made recombinants. And what we're excited about is we've gotten the HA gene from flu cloned into herpes. And when we put this into cells, we get high expression of the herpes antigen. I'm sorry, the influenza HA hemagglutinin on the surface of infected cells. So now the idea would be, can we protect against influenza? Now, part of the reason that Kayla Weiss wanted to do this is one hypothesis that you, as an immunologist, you could have asked, is if you went ahead and what we did was take out the most immunodominant antigen of herpes, maybe that's why the vaccine works. So one way we could test it is put back a very immunodominant antigen and see if it takes away the herpes protection. So I have to tell you that what we were so excited about in the last 3 months is Joe's made the vaccine. He's been looking at the immune responses in mice that are immunized with this recombinant herpes influenza vaccine. And what we excitingly see is that we get antibodies to HA that are the IgG2C, which are the ADCC antibodies. And I have to tell you, I'm new to flu. We had the first set of experiments going on about 3 months ago where we immunized the mouse. And because I didn't work with flu, I wanted to go ahead and get flu from some trusted lab. In the first experiments that we did, we got the great ADCC antibodies, but the people that sent us the flu sent us a strain that wasn't virulent. So this is this morning's result. Okay, 8 a.m. this morning, Joe is now measuring the weights of the mice. And what I want you to see is right up here, the mice that are immunized with the recombinant herpes expressing the full-length HA are protected. And at 3 o'clock this afternoon, we can even see better killing. By tomorrow, all the other mice are going to be dead. And we're super excited. We're super excited because we think we're going to have a universal flu vaccine. We're going to have to test that. But it's very different than every other flu vaccine. And by the way, if you get this vaccine, you're also going to be likely to be protected against HSV1 and HSV2. So, we're in the process of testing that. So, what I've told you is we're excited because now we know we can clone and express foreign antigens in our Delta GD2 virus and get immune responses, ADCC antibodies, to those foreign antigens. So I think the reality, the vision that I have of this universal vaccine is still one that we can hit. For the flu itself, I've gone ahead and established collaborations where we're going to be able to test against pandemic flus and really see if we have the universal flu vaccine. I have loved being here at Einstein and the collaborative atmosphere that we have here in the Einstein-Montefiore conglomerate. I'm glad that Dean Spiegel and Steve Safer have a vision of where we're going. And even though I'm going blind, I still have a vision of what we can do in the world. We're at Einstein, and Einstein said imagination is more important than knowledge. Let's always keep that in mind. And we should let our students take risks, but be committed to the long term. We want to challenge dogma, and we want to smell the roses along the way. So I am very fortunate to have this collaboration with Betsy. And we have weekly meetings, and it's a wonderful group of people. And I'm very fortunate to have a set of people working in my lab still on so many diverse projects that I couldn't even talk about today. And I just want you to see, I've had the great pleasure of having these collaborations at Einstein, and it's the Word doodle that just shows you all the papers that we've published, together, and the prominent people here from Einstein who have contributed. I'd like to thank all them, and thank you. Be glad to take any questions. Thank you.